Hi everybody, I want to give you guys a little bit of a background of cellular respiration. It can be a difficult concept to grasp. You will be covering it this week in lab. So when you say the term cellular respiration, does that mean that our cells are actually breathing? No, it means that they are using oxygen though, and they are using oxygen to make ATP. When we look at ATP, ATP stands for adenosine triphosphate, and it's kind of like this adenosine molecule. There is a sugar in there too as well, but you have 3 phosphates, and what you do is you cleave off one of these phosphates and you attach it to something to do work in the body. So you may be pushing something across the membrane, or you may be attaching it to an enzyme to do a specific task. So we think of ATP as like the money in our body. Okay, we think of it like the money we need to pay a toll to get somewhere, to move down the highway. And there are 3 steps to cellular respiration. The 3 steps are glycolysis. The second one is the citric acid cycle. It's also called the Krebs cycle after the person who discovered it. And the third one, we have the electron transport chain. Okay, now glycolysis does not require oxygen, and it occurs in the fluid portion of the cell known as the cytosol. The citric acid cycle in the electron transport chain, this occurs in an organelle called the mitochondria, which they also call the powerhouse of the cell. It's a very important organelle that's used to make ATP. We're going to show you how it does that. And we do require oxygen for these steps. It's specifically for this one, for the electron transport chain. All in all, if we just did glycolysis, we'll only make 2 ATP. If we go all the way through the electron transport chain, we can make up a total to roughly, I'm just going to give an estimate, about 36 ATP. So you get more bang for your buck when you have oxygen available and you progress through all 3 steps. There's some other terms that are used here with glycolysis. You'll hear the term anaerobic respiration. Just going to abbreviate it right here. And anaerobic means without oxygen. When plants use just glycolysis, you'll hear the term fermentation. You guys will discuss this with lab and using yeast. Whereas the citric acid cycle, because they do require oxygen, you're going to hear the term aerobic. Okay, aerobic respiration. All right, so let's go ahead and let's take a look at these steps here. All right, this is actually just an overview here. So we have glycolysis, and you can see glycolysis is occurring in the cytosol. So that is like the fluid portion of the cell. So within the boundaries of the plasma membrane, but still within the fluid here. And I'm going to get my pen ready. So you got glycolysis there, okay? And so once we enter into the mitochondria, which is this sort of pill bug-like shaped structure right there, there's a little bit of a transition between glycolysis and the citric acid cycle, but you can see the citric acid cycle right there, and then we will progress forward into the electron transport chain. But you can see these last 2 events here. This occurs within the mitochondria. You can also see below too, we have 2 ATP versus 36 ATP for these guys. So you can see you're going to get more bang for your buck when you have oxygen that is present, okay? Glycolysis is without oxygen, okay? So remember the terms we had, anaerobic and versus aerobic. All right, so let's take a look at these individual steps here. So with glycolysis, we're going to start off with glucose, and glucose is a 6 carbon molecule, and you can see that from the image right there. And you could imagine there'd be some oxygens and hydrogens around these carbons as well, but they've been omitted just because they're not necessary for you guys at this non-majors level to understand. Now we're going to be borrowing 2 ATP, so they call it the energy investment phase. We're going to be borrowing 2 ATP. So you can see you've got 2 ATP molecules, and when you have 2 ATP, remember they stand for adenosine triphosphate. So that's like saying I'm going to donate 2 phosphates to glucose, and you can see they leave 2 ATP. That stands for adenosine diphosphate. So di is 2. So that's like saying we have an ATP molecule. Remember, just 3. I might redo that again. You have an ATP molecule, which is 3 phosphates. That's like saying I'm going to donate this glucose molecule, this phosphate to you, glucose, and now I'm going to leave as ADP. Okay, so we have glucose. I'm going to be right up here now. We have glucose right here. We're going to take in 2 ATP, which will leave as 2 ADP. And now this is glucose here. You actually change the name. They don't list it here, but I'm just going to mention it, and it's not required for you to know, but you're basically going to change the name to, let me just draw my carbons here. And you're going to add your phosphates here at the end because we donated the 2 phosphate. They're going to use the term 1,6 fructose bisphosphate. So you may be reading about that. I'm running out of space over here. And the bi means 2 because you have 2 phosphates and they're located at the first carbon and at the 6th carbon. Again, not really for your concern, but you will be reading that. Okay, after this is we're going to actually split. Now the term glycolysis, glyco means sugar, and lysis means to split. So we're going to be splitting this glucose molecule or fructose molecule into 2 3-carbon molecules. And the term here that they have here is pyruvate, but you may also see the term pyruvic acid, and it is the same thing. Now there is some other intermediate steps between this. Not really of your concern here, but there is some intermediate steps that are in between, forming from fructose down to pyruvic acid. And all of these steps are enzymatic reactions, meaning that all of these steps are facilitated by enzymes. But when we did make these final 2 molecules of pyruvic acid or pyruvate, you guys can see them right there. We have 2 3-carbon molecules. When we did that, we made some stuff. Okay, and so on each side, you made 2 molecules of ATP. We made 2 ATP, not borrowed it. So we each made 2 ATP. And so you can see here, you can see a total of 4 ATP. All right. We also made NADH. Okay, we also made 1 molecule of NADH. For each side here, NADH. Sorry, my handwriting is not the nicest. And these will be used, I'm just going to put ETC, these will be used for the electron transport chain. Now, we made 2 ATP, but remember in the beginning we borrowed 2 ATP. So in essence, glycolysis only gives us 2 ATP molecules. It also gives us 2 molecules of NADH. But if we were to stop here, so let's say we don't have any oxygen, like if you were exercising vigorously, you'd be cutting off the blood flow to your muscle tissue, so therefore less oxygen delivery to that muscle tissue. We would be without oxygen. You wouldn't be making much ATP, and that's why you can only sprint or you can only weight lift for so long. Eventually you'd go through fatigue. But let's say we do have oxygen available, so we're going to progress forward. So I'm going to move on. Okay, from glycolysis, we're going to enter into what's called a transitional phase here. And you can see we've got the pyruvic acid from glycolysis. Now in this image, they only show 1 molecule, but remember there is 2 molecules of pyruvic acid, okay? And we're going to actually lose one of these carbons here. We're going to lose it as CO2. And when you breathe in oxygen, we use it to make ATP in our tissues, but CO2 is what we breathe out. That's actually a waste product from this whole process. And so if you lose a carbon from making CO2, you can now see that you would only have 2 carbons, right? Okay, so now you can see we have only 2 carbons. They did add a coenzyme, which is actually a B vitamin, coenzyme A, and that vitamins can act as enzymes. They facilitate enzymes. So you'll see the term now. It becomes what's called acetyl-CoA. So this right here, this 2-carbon molecule is acetyl-CoA. CoA. Now these two molecules are going to both enter the citric acid cycle. Ooh, my handwriting is really bad. The citric acid cycle, sorry, it's late. And the citric acid cycle, we're going to go through 2 complete turns of it because you have 2 molecules of acetyl-CoA, therefore you'll go through it 2 times. So let's go ahead and let's take a look at that citric acid cycle. And you can see here we have acetyl-CoA, okay? So we have acetyl-CoA right here. Now the term citric acid cycle comes from oxyl acetate and acetyl-CoA combining together. So you see you have 4 carbons here, you have 2 carbons here, and the first product is citric acid or citrate, and that's where this name is derived from. You did hear the name Krebs cycle, like I said before, and that is derived from the name of the person who discovered it. Now, you're going to see a bunch of enzymatic reactions, but what you're seeing here, we have 6 carbons in citrate, and you can see now we leave as CO2, so we're losing a carbon. So you can see now here we have 5 carbons. Between going from 6 carbons to 5 carbons, not only do we lose a molecule of CO2, but we also made a molecule of NADH. This will be used for the electron transport chain. Now what this NADH, and I didn't really explain this in the last one, but what this is doing is it's picking up a hydrogen. And with a hydrogen, you have 1 proton and 1 electron. So I want you to think that it's like taxi-cabbing electrons to the electron transport chain. We're going to be using these hydrogens here for energy to drive that electron transport chain. And you can see we've got another CO2 molecule leaving, so now we move down to 4 carbons. And we'll stay 4 carbons until we come back around, but we're going to be making a lot more NADH, and then what's like a twin of NADH is FADH. And these are basically just all taxi cabs. They're stealing hydrogens from these chemical reactions to use them for the electron transport chain. So that is the main purpose of the citric acid cycle, is to generate NADH and FADH, which is in these hydrogens right here. You have electrons, and that will be used for the electron transport chain. Now, you go through the citric acid cycle, you go through it 2 times. Remember, you have 2 molecules of acetyl-CoA, so you're going to go through this twice here. All right, let's go ahead and take a look at this electron transport chain. And I've used a different image because I wasn't really crazy about the one that they have in the textbook here. But remember, we are in the mitochondria, and the mitochondria kind of looks like a pill bug. So you have an outer mitochondrial membrane, and then you have an inner mitochondrial membrane. And this inner mitochondrial membrane, that is this membrane right here. Now, can you guys see NADH and FADH? Can you guys see that they're coming in as NADH, but they're leaving as NAD? FADH is coming in and leaving as FAD, so it's basically dropping off hydrogens. And what's happening here is you have a bunch of metal ions and you have a bunch of enzymes that are embedded in this membrane. You can see these large structural type of objects here. And what they are doing is they're taking the hydrogen, which has a proton and an electron, okay? And they are pumping the protons out there into this space. Okay, but then the electrons, can you guys see that little yellow arrow? I'm going to kind of clean this up. Well, let me change the arrow here, color. So you guys see, oh, it's changing all the color. Sorry about that. Let me change it back here. So you guys can see all these yellow arrows, and this is the flow of electrons. All the electrons are going to be flowing in this inner mitochondrial membrane. And let me just kind of draw it out here. So this is the inner mitochondrial membrane right here. And you're going to have FAD, H and NADH. They're dropping off the protons. And what happens is that you have proteins that are embedded and enzymes. Well, enzymes are proteins, but enzymes and metal ions that are embedded in this inner mitochondrial membrane. And they're going to pump the protons out there, and the electrons are going to be shuttled in the inside here. And it's going to create a gradient, kind of like an opposite end of a magnet pole, but they can't touch each other, but they want to be by each other. And the electrons will actually be drawn, so they'll kind of shuttle along that inner mitochondrial membrane, and they'll be drawn to the water molecule. So water is actually, I'm sorry, let me repeat that again. Just ignore that last step there. They will be drawn towards the oxygen molecule. So remember we said oxygen was required, okay, for this process. So where is it actually required? Now when we see O2, that's oxygen gas, but oxygen can actually be by itself. And when it is by itself, I drew a little negative sign, it's very electronegative. So oxygen is where the electron party is at. So all of these electrons are just going to be drawn towards that oxygen. And these positive protons out here are going to be like, oh my God, I want to be there. Oh my God, I want to be there. And they're just going to be creating this sort of energy sort of gradient, okay? Now there's one point where these protons can pass through, and this is through a molecule called ATP synthase. And when these protons rush through this protein channel called ATP synthase, they sort of like a turbine, you know, when water flows through a turbine in a dam and that generates electricity. Think of it like that. The protons run through this channel here and causing this protein channel to spin. And when it spins, it generates energy, okay? Now, I'll tell you what that energy is to do in just a second, but that proton will join with the oxygen and we'll have water. And one of the results of cellular respiration, or I should say the product, is water. Now while these protons are running through here, this is spinning and spinning and spinning. And it's spinning, it's taking an ADP, which is kind of like a used ATP, combines it with a free floating, that little I stands for an inorganic phosphate, and we get ATP. And this is how we make ATP. A boatload of it, okay? All right, so that is in short the overview of cellular respiration. I'm actually going to go back to the first page there and give you guys the equation for cellular respiration, even though you're talking about it in your class. But the equation is C6, that's a C6H12O6 plus O6. Oh, I'm sorry, hold on a second. 6O2 is going to give us 6CO2 plus 6 water plus, I'm just going to give you a number, but this number can range 36 ATP. Okay, so what this is saying here, and you guys see how you have 6 carbons over here and see how you have 6 carbons over there. This is saying that we have glucose. And in the presence of oxygen, we are going to form carbon dioxide plus water. Well, you guys know what H2O is, water, plus ATP, which is energy. So this is the overall equation for cellular respiration. And you guys, like I said, will be going through this and over this in lab. All right, thank you very much.